Hello everyone, this is lecture first for the bacterial structure. And in this lecture we are going to understand size and shape of bacteria. Let's start our lecture. So see this figure of bacterial cell where we can observe the different parts. Now see, this is the nuclear, which is the DNA, that may be RNA also. This is the plasmid, which is extra chromosomal DNA, and this is a flagellum, which is helpful in the locomotion purpose. This is a glycocalyx. It is also called as a capsule. This is a ribosome, which is helpful in the protein synthesis. These are the inclusion. This is a mesosome, which is helpful in the reproduction. And this is the cytoplasm. This is the plasma membrane. This is the cell wall, and these are the pili. So this is simple structure of bacterial cell. Now see, this structure is divided into two parts. First one, structure which is external to the cell wall, which includes flagella, pili, which is called as fimbriae also, capsule, sheaths, prostheses, stalks, and the cell wall. The structure internal to the cell wall, which is cytoplasmic membrane, intracellular membrane system, cytoplasmic inclusion and vacuoles, and the nuclear material. So these are the two main types of structures which is divided into external to the cell wall and internal to the cell wall. Now let's see first one, that is the size. Now in this video lecture we are going to focus topic of bacterial structure on the size and shape. So a bacterial cell is unicellular and it is a microscopic in nature. The average size of bacteria, which ranges in between to the 0.2 to 2 micrometer in diameter. Bacteria such as surgeon fish, symbiont, Epulopiscium, fishelsoni are very large in size. The size is around 80 micrometer in diameter with more than 0.6 millimeter in length. The sulfur chemolithotroph Thiomargarita namibiensis, which was discovered in ocean sediments in Namibia, that is Southeast Africa, may grow to be as the largest bacterial cell around 0.75 mm in diameter. So the largest bacterial cell is the Thiomargarita namibiensis. Ultra small bacteria have been discovered in open ocean that was passed through a filter with a pore size 0.2 micrometer. These bacterial cells are also called as a nanobacteria. They contain DNA, an average of 42 ribosomes per bacterium, and in possession with the pili. See, for example, Escherichia coli, which the size of Escherichia coli is in between the 1.1 to 1.5 micrometer in diameter by 2 to 6 micrometer in length. Spirochaetes contain size like 500 micrometer in length. Cyanobacteria oscillatoria contains 7 micrometer in diameter. Mycoplasma, which is also called as a PPLO, the size is 0.25 micrometer. Mycoplasma galliseptum, with the size of approximately 200 to 300 nanometer, are thought to be the world's smallest bacteria. Now let's see another point is the surface to volume ratio. Now see, as far as the bacterial size is concerned, the most important thing is surface to volume ratio, and that point we have to consider during the during the measurement of size of bacterial cell. See, the most important consequence of the small size of microorganism is that the surface to volume ratio of bacteria is exceedingly high compared with the same ratio for larger organisms with similar shape. Now see, the surface to volume ratio of bacteria is very high as compared to the other microorganisms. Consider the simple case of sphere in which the volume is a function of cube of the radius. Then it will be V is equals to 4 divided by 3 pi r cube. While the surface area is the function of the square of the radius, that is S is equal to 4 pi r square. Now, a cell with a smaller r value therefore has a higher that is the surface area to volume ratio than a cell with the larger r value. In the process, they are able to continue growing and reproducing as a steady rate. Now see, the size of bacterial cell is very small. Very small, and that's why there is surface to volume ratio is very high. And due to that surface to volume ratio, the bacterial cell is able to grow as far as possible, and that's what they are able to take nutrients very fast as compared to other organisms. So they are also able to distribute that nutrients, whatever nutrients they are taking inside, into the body as far as possible as compared to other organisms. They will do the very fast metabolism because the cell is very small. Okay, so there is a no requirement of like large time or like that removal of waste material. So they will remove the waste material very fast as compared to other organisms because they are very small. Next point is the shape of bacterial cell. Now see, first shape is the cocci. Cocci means the circular shape. Example is the micrococcus aureus. Then bacilli, this is the rod shaped bacteria. Example is the bacillus cereus. Spirilla shaped bacteria are like spiral shaped. Example spirilla minis. Okay, this is the cocci, this is the bacilli, and this is spiral shaped bacteria. Comma shaped, the bacteria are like comma shaped. For example, Vibrio cholerae. Pleomorphic means the bacteria don't have their specific shape. They will change their shape on the basis of their conditions. Those are known as pleomorphic bacterial cell. Example, rhizobium. Star shaped bacteria means the bacteria are like star shaped. Example, Stella humosa. This is the example. Rectangular shape, they are like box shaped bacteria, are rectangular in shape and resemble a box. Example, Sarcina luta. This is the example, Sarcina luta. So this is all about the bacterial size and shape and their basics. Thanks a lot.